Hello, we're glad you've joined us for this live webinar, The Molecular Analysis on Circulating Tumor Cells to Determine Prognostic and Predictive Factors in Cancer Research. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by QIAGEN. Whether your passion for answers takes you to a laboratory bench or a hospital bedside, the pursuit often begins with a blood or tissue sample. The deepest mysteries of disease and other biological processes are encoded in the building blocks of life, DNA. Working with these molecules is quite a journey, and we at QIAGEN understand these challenges. QIAGEN offers you innovative solutions that cover every single step along this journey, not just technologies, but bridges from samples to insight. Samples to Insight means QIAGEN offers you the industry's most reliable sample technologies because samples matter to your success. Our top quality assays and panels enable you to accurately analyze and identify diseases and genetic variations. Our bioinformatics software and curated knowledge bases transform your raw data into relevant, actionable findings, and our automation solutions provide you seamless and cost-effective workflows. Sample to Insight. The insights you gain may lead to one small step or a giant leap forward for science and healthcare. Partnering with QIAGEN, your work can make a difference. Now let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Siegfried Hauch, PhD, the Director of Liquid Biopsy and Site Manager at QIAGEN in Hanover, Germany. Dr. Siegfried Hauch studied biology at the Albert Einstein University in Ulm and holds a PhD in plant physiology and molecular biology. He served as Director of R&D at AdnaGene since 2006 to build the AdnaGene Circulating Tumor Cell business. In 2015, Dr. Hauch was appointed as Director of Liquid Biopsy and Site Manager at QIAGEN Hanover GmbH. Now he is responsible for the Circulating Tumor Cell business at QIAGEN. Dr. Hauch will now begin his presentation.
Thank you very much for this long and kind introduction, and thank you also very much to the auditory that dialed in as far as I know worldwide. So I say hello, good afternoon, good evening, and good morning to the auditory. So talking about liquid biopsy, that's a very interesting and a very challenging field in the moment, and there is many approaches actually how one could address that. And we really have to focus today to the circulating tumor cells only because I could talk for another 3 hours about all the rest. So forgive me that it's only about CTCs today, but I hope I can kind of convince you that it's not this is CTC content that we have here, what we can learn from the circulating tumor cells, contributes so much to the liquid biopsy field that finally we would not like to live without it. So my title is here, Harnessing the High Value of Circulating Tumor Cells in Liquid Biopsy. And before I can really start, I would like to bring up this legal disclaimer here, telling you a lot of things, but mainly that we are talking about research today. We are not talking about the clinical routine and the clinical diagnostics. It's only about research and the use of circulating tumor cells as a part of the liquid biopsy challenge in such endeavor. So let me see. Before we can deeper dive into that, I would like to introduce you to the test system that we developed and used in that context in our research work, and this is the AdnaTest principle, which is actually an easy principle on one hand, but a tricky one on the other hand. So what do? You may have heard, if you are in that field already, you may have heard about immunomagnetic cell enrichment, which is quite commonly discussed in the field of CTCs over years, and also the use of transcriptome profiling to characterize the CTC. So what is different with the AdnaTest principle here? And this is the so-called, what you call COCP principle. It means combination of combinations principle. And it starts first with an immunomagnetic selection or enrichment of the cells that is not as common as you may think. So we do not use a single antibody directed against a single tumor antigen to enrich the cells from the blood, because we learned very early, even in 2004 already, that once the CTCs appear in the blood, in other words, once a tumor disseminates CTCs into the bloodstream, there is a dramatic change in phenotype and profile of the cells to make that happen. Using only a specific antibody that might be expressed on the cells in the tumor tissue can easily be, and that is easily be lost during this transformation process. And if you use such a kind of an epitope for selection, you might not find your cells anymore and might get false negative. The part here, how we dealt with that issue was that we use a mixture of different bead populations containing different antibodies direction, again, various epitopes on the tumor cells, all in order to kind of being prepared that if one or two of them is lost, there is still an epitope available that can be used for will be used for the enrichment. Doing so, we are quite successful and have very high sensitivity in the CTC capturing, which is due to that effect. That is then followed by the start of the 8-mRNA isolation and reverse transcription to get cDNA. Important to understand here that we are not visualizing the cells, we immediately lyse the cells, because we say that the cell as such does not contain or is not the important information. The important information that we want to explore is what is in that cells. What information can we get out of that? And this is much more than only knowing that they are there. And the second combination of the combination of combinations principle is then multiplex PCR approach, again following the same idea that the expression profiles in circulating tumor cells might be highly variable, and therefore you need to address several tumor markers on the RNA basis to make sure that you may not miss the cells or may not get negative findings only because of one or the other of the markers is not overexpressed in these cells. The rule is here for the detection of CTCs, if one of the markers we have there in the multiplex PCR reaction is getting positive, this is counted as a proof for the presence of CTCs. So this is about the COCP principle and how it works, and how does that look like in practice. So the AdnaTest is a benchtop assay. There is not much automation on it. Actually today there is no automation on it. It's a manual test. It's a very straightforward procedure. You start with 5 milliliters of blood. You incubate the blood with the beads and for 30 minutes at room temperature. This is followed by several washing steps actually. After all these washing steps are done, you have the enriched tumor cells there. To give you an idea about how much the enrichment or how we can kind of quantify the enrichment quality is, that in the normal AdnaTest assays we have then about 1,000 leukocytes still contaminating the CTCs, but this is already good enough to go into the RT and multiplex PCR reaction with a very high specificity depending on the markers you are analyzing here. There are already several tests available, commercially available in the market. That is a test for breast cancer, for prostate cancer, the AdnaTest colorectal cancer, the AdnaTest ovarian cancer, and 3 more assays that are mostly used then in the research field today. This is the AdnaTest ovarian cancer 2 that deals with EMT-like cells in the ovarian CTC field, and the AdnaTest EMT1 and EMT2 stem cell that is dedicated to not only address epithelial-like cells in the blood, but also cells under EMT, epithelial mesenchymal transition, or cells that have a tumor stem cell-like phenotype, in order to also identify such cells and look what kind of metabolism is behind such cells. Okay, enough about the test. So I hope I could briefly describe you what kind of approach we used here for what I have to tell you now. Because what are we really asking from CTCs? Did we place the right questions? And did CTCs tell us all they could so far? This is what it's about. And if we are now starting with the prognostic information that we can obtain if we look at CTCs, this is only one side of the coin, actually. So it's an important side of the coin, but finally not enough and not leading to the final decision-making, something that a doctor may need to change his treatment to treat the patient better. Nevertheless, using the technology I described to you, there are several papers out there where we really can show this is confirmed. There is prognostic information in the CTC content, in the CTC presence, if circulating tumor cells are there in the circulation, either after surgery or during a therapy, and the therapy is not removing the cells from the bloodstream, then this is a bad prognostic factor. Published, I gave you here a few publications. There are much more, but I think this is kind of initial publications we made in that context for several tumor entities. This is true for breast cancer, colon cancer, for prostate cancer too. We found it in ovarian cancer, and also applying the EMT1 and stem cell-like tests. We found that also for the tumor stemness-like cells and the EMT cells in human breast cancer. What does that mean? I show you here a few results. So this study was made with Sabine Casimir-Bauer from the University of Essen in 2014, published at the San Antonio Breast Cancer Symposium in that year. That means that circulating tumor cells in primary breast cancer, for instance, if they are there after surgery, this patient population shows a worse disease-free survival, actually, compared to the patients where the CTCs were not detectable after surgery. In principle, that's a good idea because it could help us to divide the patient cohorts into subgroups. One that is under much higher risk, and one that has a rather low risk to form metastasis, really. There is only one problem with prognostic information. And that is the question that always comes from the oncologists to me. But do I have something what I can do to help the cohort that is under risk? And this is a very, very important question because it cannot be really answered until now, except if you see what we found in terms of predictive information. Because that is really where we have to come to. The prognostic information is nice, but we have to tell the doctors or give the doctors the information what to do now with their patients. One could say in breast cancer, okay, observe them better. Give them longer treatment of whatever you give. Adjuvant, even a second adjuvant therapy could be discussed in that context. But still the question remains, will that really help? And this is the question that we have to answer. Nevertheless, you can see here, again, the same story in breast cancer in the metastatic situation, even more clear. If CTCs are there under therapy and they stay there and the therapy is not able to remove the CTCs from the blood, the patients have a much shorter overall survival than the patients where this is not the case. Important in this study done by Teves et al. In 2009, important here is that all these patients had a very long therapy history. We are not in third line, we are even behind third line here. And even though these patients, if the CTCs get removed by the therapy from the blood, the patients have a much longer survival than the patients where this is not happening as a consequence of the therapy. In ovarian cancer, as you can imagine, it's even more clear. Ovarian cancer before and or after surgery, the CTCs have a even before surgery, the CTCs have a very clear prognostic information. If CTCs detected by the AdnaTest ovarian cancer are already there before the surgery, these patients show a much shorter overall survival compared to patients that were CTC negative before therapy. And this is even more clear for the disease-free survival. After roughly 30 months, all the patients that had CTCs before therapy relapsed, whereas at least 40% of the patients had no relapse until 60 months or so. A very clear information that we get from the CTCs as such. These patients have a much higher risk for metastasization in ovarian cancer. But again, the question, can we do something against that? Do we have more information of the CTCs? Can we get out more information of the CTCs we analyzed here to help these patients really? Not only to tell them, okay, you are a risk group now, your chance to develop metastases earlier is probably higher, but we have no therapeutic option to treat them better, to give them a better chance and to help them better. The same you can see also in prostate cancer. We did here a study with Totenhöfer et al. In 2012, where we can clearly see that CTCs even give us an idea about therapy response or not. You can, in that study, the CTC negative group, the group under remission, never showed CTCs before therapy. They did not show CTCs during therapy. Or few CTCs during therapy and never CTCs after therapy. The other situation we see in contrast in the progressive group, here we do always find CTCs either before therapy or after, but they were never CTC negative as such. So it's a good discriminator. It's a good discriminator for therapy responsiveness, but we have still not an idea about what to do now as a consequence of these findings. And therefore, I bring you now to the most important chapter of this talk. CTCs, do they have the potential to give us predictive information? And 3 of the papers I already mentioned for the prognostic value may be chosen here in colorectal cancer, in prostate and ovarian cancer, where we really could find such kind of predictive information, and there is even one more in breast cancer, which I will bring you as a highlight in the end of the presentation. Let's look to colorectal cancer. We found, indeed, that circulating tumor cells are, again, prognostic, but also in a group that has wild-type KRAS, and is therefore treated with cetuximab and panitumumab. Musella et al. In 2015 in Milano, they observed that finding in their trial that CTCs, or that there is a certain group of therapy, generic therapy non-responders to anti-KRAS therapy, and they were asking themselves, why is that? And they used CTCs as a potential explain for what could happen here. And indeed, what you can see, even progression-free survival, but also overall survival, is clearly correlated to circulating tumor cells in colorectal cancer patients. The question comes now up, why is that so? Why are these patients not responding to a therapy even if they wild-type RAS-RAF patients? It's not flipping forward. Let me see what I can do. So, and You can even drive that further. You can even make it more stringent and analyze that in favorable and unfavorable, but nevertheless it's always the same message here. Circulating tumor cells are the reason for or seem to be, I must be scientific here, seem to be the reason for panitumumab failure in KRAS wild-type patients. And there might be a certain explanation for that, and this is presented here. In another trial we did with the group at Policlinico in Rome around Raimondi et al. In 2013, we analyzed the CTCs and compared them to tissue wild-type and tissue mutants, and we added to the normal colorectal assay another molecular assay testing for KRAS overexpression. And this is the interesting result we found here. In the tissue mutated patients, none of them, even if it's a small cohort here but highly significant, in the tissue mutated cohort, none of them showed CTCs with KRAS overexpression. But in the tissue wild-type patients, the CTCs that we found there were in 50% of the cases showed a KRAS overexpression signature, which could be fairly explained that we might be able to identify anti-KRAS panitumumab treated patients that will not respond to the treatment, not because of they have a KRAS mutation, but because of they have another amplification of the KRAS that leads to the same effect, and this is the KRAS overexpression. Another picture to the same story, and already kind of leading into the right direction. Here we can predict at least...
वो डॉक्टर वही बोला उसको कि तेरा ऑप्शन है. I don't want to jump into something unless he's hesitant. बोलता है मेरे को करने का नहीं है, लेकिन मेरे को I don't think you'll have problems justifying it at the insurance level because of the तेरा ये bleeding. This is not right, this level of bleeding, so that's not a problem. You tell me कि तेरे को निकालने का बोलेगा तो निकाल सकते हैं.
We haven't. There is somebody in the line. Okay.
Yeah, I'm here. Yeah.
No, that's why हम लोग सब लोग को ये प्लान किए, सोच रहा कि ये ऐसा करता हूँ, ऐसा नो वी शुड नो. एनीमिया का उसको डर है, इसके लिए डॉक्टर बोला कि एनीमिया अभी तक आया नहीं है, तेरा ठीक है अभी, लेकिन उसको और
I cannot understand what is said really here, I must say.
पट करके आएगा, that is the problem with that, एकदम धड़क करके आएगा.
Okay, go ahead. I will talk to him. Sorry about that.
Okay, so what I try to explain you is the following thing, that there is more to see, but the important message is the right way where we should focus probably. We should focus to a therapy prediction. So what we can see here really in a certain therapeutic regimen can predict that this therapeutic regimen is probably effective or not. This is already a kind of prediction, I would say. It is more, at least, than prognosis, but I will show you more that it goes even more in the direction of prediction. And this is here on my next slide. You have seen the prognostic effect of CTCs in ovarian cancer, but more than that, we tried to find predictive information in these cancer cells, and therefore we were looking for genes that could predict kind of something against the normal therapeutic regimens you give in ovarian cancer. And one of them is first-line platinum therapy. That is a very common therapy form that usually ovarian cancer patients get at the first instance. And we analyzed a special gene in top of the genes that are normally in the ovarian cancer test, and this is the gene ERCC1, and we tested the overexpression of ERCC1, which is DNA, it belongs to the DNA repair gene family. We chosen that because of platinum is working as a DNA damaging therapy in tumor cells, and therefore the overexpression of a repair gene could fairly lead to platinum resistance. And indeed this was the case. CTC, and you see that in the last line of this table here, CTCs positive for ERCC1 were not only highly and independently prognostic, even to be demonstrated in the Cox regression for disease-free survival. And when I say independent, I mean independent of the normal staging factors, actually. But we found also them to be an independent factor for platinum resistance. In other words, if we see patients before surgery, I should say that before surgery, we analyze the patients for ERCC1 positive CTCs. If we find them, it is most likely that these patients will not respond after surgery to the adjuvant platinum first-line therapy. This is already coming very close to predictive information. The only what we would like to provide now is an alternative, but this could be already second line or kind of paclitaxel-based combination therapy, or even kind of personalized therapy with the upcoming olaparib, which is quite under discussion in the moment. And this story, some of you may already have heard. This is one of the latest proof that we could deliver in terms of predicting therapy failure, really, before the onset of the therapy. A group around the Johns Hopkins research guys, Anton Arakis and Yun Liu, analyzed using the AdnaTest prostate cancer an additional gene for its expression profile, and this is the androgen receptor variant 7. And what they found was super interesting. They found if the prostate cancer patients, I should say here castrate-resistant metastatic prostate cancer, if they were had positive CTCs for ARV7, so if ARV7 was overexpressed in CTCs found there, these patients absolutely did not respond anymore to abiraterone or enzalutamide. This was a super clear message here. ARV7 as a self-constitutively activated variant of androgen receptor is making the enzalutamide and the abiraterone therapy useless. This could save a lot of money, this could save a lot of time to these patients if one would switch to an alternative form. And the same group in the year after, the data are not shown here, but the same group in the year after could really demonstrate also that instead of giving them enzalutamide or abiraterone, these patients were very nicely responding to paclitaxel, which would be in this case a super good alternative to a treatment of which we know, based on the CTC information, that these therapeutic regimens will not work at all. And now, as promised, I come to the last chapter in predictive information, and this is about breast cancer. And it's about CTCs that we find there and analyzed for HER2 overexpression and what this may tell us. The idea to that came from a piece of research that we did long ago. It was in 2006, I think, or so, when we compared in primary breast cancer the HER2 status or the HER2 mRNA status in CTCs and compared that to the HER2 status in primary tissue. And the interesting finding was here that only in 50% of the cases we had concordance, and in another 50% of the cases we had discordance between tissue and the CTCs. 21% of the HER2 positive CTCs were negative in tissue for HER2, and 30% of the tissue positive CTCs were negative for HER2 expression in the CTCs. So seeing that, we thought, wow, that's an interesting story, and but is that real? Is that real? Has that a consequence? And indeed, it has a consequence. We had the chance in 2013 to compare metastatic tissue with the CTCs we found there directly. And here we found another story. The HER2 expression on the CTCs in the metastatic situation was clearly correlated to the phenotype of the metastatic tissue. It was not easy to get, because you know that all. Metastatic tissue is difficult to get and difficult to obtain, but in these cases where we could make that happen, we found indeed there is a good correlation to the metastatic tissues if you only analyze the CTCs. This is something where I would say a very good example what really liquid biopsy means, because you have an easy access to that, it's minimal invasive, but you get information about the metastatic phenotype, the metastatic tissue phenotype, and again, probably an indication how to treat these patients then, because if this is real, this metastasis and this metastatic tissue should, based on these results, respond to an anti-HER2 targeted therapy. Interestingly, by the way, we did not find that really in when we stained the CTCs using the cell search approach for HER2 positivity. We found no correlation. We were asking ourselves, why can that happen? And finally, we found out a potential reason for that, because if we were looking to the CTC or to the patient we found here and the CTCs analyzed using the AdnaTest, we found that 50% of these CTCs lost AbCam, but instead of overexpressed HER2. This might be an explanation why the AdnaTest using a multi-antibody capturing approach still captures these cells and therefore gets that information, whereas the cell search approach only dealing with AbCam was probably missing the cells of interest, and therefore there is no correlation here to the metastatic tissue. Could be one explanation. One should deeper look into that to confirm it. Based on these results, we thought we go now one step further. We want to have more information about the metabolism that is ongoing here, and therefore we applied to the cDNA we got from such patient samples a whole panel of genes of interest that we wanted to see during several changes of therapy in follow-up. The genes we were looking at, you see here on the right-hand side, this is HER3, HER2, ALDH1, AbCam, c-Met, aurora kinase, PI3K, ERCC1, which we know from the ovarian because of platinum resistance, a few controls like CD45 to somehow get an idea of how many leukocytes interfere here, GAPDH as a marker, and also the EMT-related AKT2 and another tyrosine kinase, EGFR. The design of the study was like follows. We enrolled the patients under progression and at the time point T0. From there on, they got a standard therapy, which we did not interfere with what the doctor said, but after 8 to 12 weeks or so, we kind of staged the patients in a non-responder group and a responder group. Again, we took a sample and analyzed for CTCs and for that panel. And then in the non-responder group, they switched the therapy to an alternative. In the responder group, they stayed with the same therapy as before. After another 12 weeks, we again grouped them and staged them in overall non-responder group, in a late responder group, in a late non-responder group, and into an overall responder group. We got many results out there, and the paper about that is already under, it's accepted for publication, should be out in probably a month or two from now with all the detailed results. To not steal too much of your time, I give you only here a kind of a summary of the most important findings, I think here. And this is comparing now the overall responder group as an example with the non-responder group over all the time points, T0, T1, and T2, for 4 dedicated genes. The first is AbCam, where we can clearly see that you see that in red, that in the overall non-responder group, we start at time point T0 with an frequency of 50%, means 50% of the CTC positive patients there were positive for AbCam2, and over the course of the therapies in the non-responder group, this was increasing all the time until nearly 80% or so. Whereas in the overall responder group, the level was decreasing over the course of the therapy from 40 to roughly 30%. The same for aurora kinase, which we've chosen also as a kind of a marker for therapy resistance. And here we also see that in the non-responder group, aurora kinase seems to be very much correlated with that, starting with a frequency of 30% in the beginning at T0 and increasing to up to over 60% at the time point T2, whereas in the overall responder group, the frequency of aurora kinase positive CTC stayed the same or even was decreasing a little bit. But now to the most important ones. Look at HER2, and again think about what we said about HER2 the last two slides. We see at time point T0 a positivity rate, a frequency for HER2 around 10% for both groups. But in the non-responder group, the frequency of HER2 overexpressing CTCs was raising to 40% and did not decline again, did not decrease again at time point T2. The important thing to say here is what we are talking about here is really newly acquired HER2 positive. These patients did not have HER2 positive CTCs before. They did, in most of the cases, not even have a HER2 positive tissue before. This is HER2 that comes as a reason of therapy resistance, as I can see that. So, whereas in the group of the overall responders, you do not see that effect. It also starts with probably 10% frequency in the beginning at time point T0, but nearly gets to 0 at time point T2. And in combination to the HER2, another very interesting gene, the HER3, which is an actually inactive transcription factor, inactive tyrosine kinase, but that is able to activate HER2 and EGFR by dimer formation. And also for this HER3, we see the same phenomenon, starting in the non-responder group and in the responder group at 10% frequency at time point T0, and then increasing dramatically to nearly 50%. So every second of these patients was then positive for HER2 in addition, whereas it stayed roughly at the 10% level in the overall responder group. Taking this together with the findings that we had before, I go back and show you clearly what I mean with the findings that such CTCs are correlated to the corresponding metastatic tissue in terms of their HER2 profile. For me, this study clearly showed really that we have a group of patients here that probably could be a very good candidate for anti-HER2 or anti-HER3 directed therapy as a predictive consequence of such findings. So we are very close to the end of the session today. Only 2 slides. A summary. The molecular detection of circulating tumor cells using the AdnaTest has been proven and published and peer-reviewed to be a very highly sensitive and a super specific method. If I say specific, I'm talking about a specificity of 95% or better to identify CTCs and to analyze them for molecular content of the CTCs. In other words, we open the box and look into it, and we can get prognostic and predictive information out of that. Using the AdnaTests, we so far could show that they are able to monitor therapy efficacy and to determine that right after the beginning of the treatment, about in cycle 1 or so, means very early in the treatment, giving probably the chance then to make changes of the treatment if there is an indication to do so. But even more, the last 2 points are the most important, I think, in that context, what I wanted to tell you now, today, and that is that the molecular characterization of CTCs has, for me now, proven that it shows EMT and tumor cell stem cell related phenotypes, and they are linked to resistance to conventional therapy, asking for alternatives here. And the AdnaTest is an open platform, and this is an important information for you researchers in the auditory. It's an open platform that enables the investigation of characteristics or profiles in CTCs based on your research that you are actually do. You are not limited to the content that we offer with our kits here. Is wide open, and you can analyze whatever profile you may want to analyze to probably even find such interesting predictive information, at least a hint for such predictive information in your own research. Having that said, I come to the end. I hope I could give you a little idea about the interesting field CTCs in the whole context of liquid biopsy, which is also circulating free DNA, exosomes, and whatever we can imagine to have there. Everything is of importance. Everything in that liquid biopsy field can bring good information to help the patients further and to help the doctors to treat the patients further and to make them a longer life. And this is what it's all about. Thank you very much for your attention.
Thank you, Dr. Hauch, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Hauch will answer as many questions as time permits. And the first question is, in regards to the acquisition and management of CTCs, how can we better determine whether the acquisition of CTCs influences their behavior or molecular profile at the time of the assay?
Yeah, that's a question I hear rather often, and it has to do with sample preparation. So what needs to be done to guarantee a certain stability of the CTCs from blood draw to lab bench, you need to stabilize these CTCs in a way, and this is not easy to do, really. You need to do it in a way that these cells maintain viable. They should not change their expression profile and until they arrive to the lab bench. We found two solutions for it. One solution is the Adna Collect tube that we distribute in Europe, which is CE-IVD approved and can be sold in Europe, but not in the United States. For the United States, we found another strategy that uses BD vacuotenors, the ACDA BD vacuotenors, compared with a kind of an in-lab stabilization that has to be done before you incubate with the beads, and this at least guarantees a stability of what we are looking here, and we have proven that in spiking experiments, but also in patient samples. It guarantees a stability of about 36 hours, not more, unfortunately, but 36 hours already is, I think, time enough to get the blood samples from the bed to the bench site.
What role does EMT play in CTC, and how does it affect the analysis of CTC?
The role of EMT is a big role. I didn't mention too much in this presentation about that, but we have other webinars upcoming where we go deeper into the EMT field. CTCs, to be able to escape the tissue, must be transformed in a way. See, they must get from an epithelial cell that is not able to migrate to something that can migrate, and therefore people say this is a mesenchymal cell, like something. And the process that leads to that phenomenon is the so-called EMT process. So there is a phenotype change that allows the cells at least to escape from the tissue and to get into the blood vessel by overexpressing or downregulating certain genes that are necessary to get out of the tissue, really, like downregulation of E-cadherin, for example, and the upregulation of N-cadherin, and to even break through the blood vessel and get into the circulation. Examples for that are manifold. We found that, for example, in the breast cancer field, genes involved here are AKT2, the PI3K pathway, PTEN, mTOR, but also certain other pathway shortcuts over EGFR, but leading always into this AKT area. There is other indications in colorectal cancer that it's KRAS that plays a role here, and we had findings in lung cancer that c-MET is the driver of EMT in c-MET. Okay, now the cells are in the blood. And how do these cells survive in the blood, really? So because usually body defense is very good, and you should kill cells like that. And here comes over the EMT also the CTC tumor stemness phenotype into the play that makes it possible that CTCs mimic themselves to be stem cell-like. They downregulate MHC class proteins. They upregulate PD-L1 or kind of anti-T cell proteins on the surface to be invisible for the immune system. So EMT in CTCs is a very common, very necessary, and very often observable process.
What is the best method to use in rich CTC for mutation analysis?
Oh, that's a good question. So for mutation analysis, CTCs are usually, and this is my personal opinion, a tricky sample to use. And I would say it's not a matter of the enrichment process really, it's a matter of the either or low content of DNA that we have here. If you think about that, we usually have frequencies of 10 CTCs in a 5 ml blood draw. Here I would say we should probably more focus on circulating free DNA to address this mutation issue, at least on the at the time being. We are working on mutation analysis in CTCs indeed, but it's a tricky thing and needs a lot of optimization and good ideas in the future. Give me another 2 years, then I'll tell you more about that.
Will you optimize the AdnaTest system in such a way that it will be possible to enrich CTCs so that they can be cultured?
Yes. I know, even using the AdnaTest, that several groups around at the university in Prague use the CTCs enriched by the labeling method, by positive selection using beads, and they were able to bring these cells in culture. One could probably think about also label-free methods could have an advantage over the labeling, the bead labeling CTC isolation processes. I have no clear opinion about it. It's not very often done. I did not see too many papers or research about that. But what is there another issue that one should take into consideration? Amongst the very limited research I have seen so far in that context, there seems to be also one message, and this is that it's not, even if you get them viable and label-free, it's not easy to take the CTCs to get them to grow. Because if they are this EMT-like phenotype, there might be even a growth inhibition in such a situation, and therefore it's not easy to get them really in culture in that situation. And if they then grow, the question is, are they still the same than they were before?
Well, thank you for that. I would like to once again thank Dr. Hauch for his presentation. Do you have any final comments?
No, actually not. I'm happy. One comment probably, I hope I could kind of trigger the interest in the auditory to try to get more information about CTCs, try to do research in the CTC field. From my perspective, I can say it's super interesting. There is so much information we do not have yet, and again, one thing to remind you, what we are doing that for is for the patient's survival and for making them a longer and better life.
Great. Thank you. I'd also like to thank the audience for joining us today and for their interesting questions. And we would like to thank our sponsor, QIAGEN, for underwriting today's educational webcast. This webcast can be viewed on demand through November 12, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Bye-bye.